Good morning, sir. Good morning, madam.
Good morning, sir. Be like a proton. Stay positive. First of all, I would like to thank you all for joining us. The purpose of organizing the webinar on the discovery of medicine is to educate and inform individuals, particularly those in the healthcare and pharmaceutical industries, about the latest developments and innovations in the field of drug discovery. Webinars can provide a platform for experts in the field to share their knowledge and expertise with a wider audience. They can also provide an opportunity for attendees to ask questions and engage in discussions about topics related to drug discovery, such as the latest trends in drug development, new techniques and technologies for drug discovery, and the challenges and opportunities facing the industry. Overall, the webinars on the discovery of medicines can help to advance the field of drug discovery and ultimately contribute to the development of new and more effective treatments for a variety of diseases and conditions. Let me take the opportunity of our dynamic principal, Dr. RAJAMAHENDRAVARAM, who is a friend, guiding spirit, and a woman of strategic wisdom and true leader. I feel it like a responsibility to thank her for her guidance and support in organizing the international webinar by the Department of Chemistry. Now I request Dr. RAJAMAHENDRAVARAM, Madam, to give the opening remarks of the international.
Thank you, Sunita, head of the Department of Chemistry, for giving me this opportunity to participate in the international webinar. Distinguished speakers and esteemed participants, a very good morning and warm welcome to you all. It's my pleasure to begin this webinar, Discovery of Medicine. I take this opportunity to thank our Commissioner Sir and CCE Authority for permitting to conduct this webinar, international webinar. I appreciate the Chemistry Department for taking initiative to organize this webinar. Congratulations, Department of Chemistry Organizing Committee. On this occasion, I would like to speak about our college. Srimati Kandakuri Rajalakshmi College for Women, Rajamahindra, was established in the year 1968. In the name in memory of wife of Rao Bahadur Kandakuri Veeraslingam Pantulu Garu, a famous social reformer of South India and a philanthropist. Sri P.V. Narasimha Rao, the then Education Minister and also former Prime Minister of India, laid the foundation stone. We successfully completed 54 years qualitatively and quantitatively both. Golden Jubilee celebration celebrated in 2018. Our college was running as aided college, but recently it has been taken over by the government of Andhra Pradesh as per UIMS number 28, dated August 10th, 2022. The college was reaccredited at B+ level with NAAC. So that in this connection, we received ROSA funds of 2 crores of rupees. We are offering conventional restructured courses in UG, besides MSc Zoology. Our college was born with a noble mission and to impact quality education through effective pedagogy for exploration of knowledge, reflecting innovation, creativity, and lateral thinking, and also to stay the young girls towards expansion concern for the reservation of our heritage and culture and conservation of environment. The vision of the college is to transform the young women into integrated, empowered, and holistic women leaders with a sound physical, mental, and spiritual facilities who are service-oriented, environment-conscious, and proactive to the development of family, society, and nation. Today's topic is discovery of medicine. It is a complex and challenging process which involves identifying and developing new medications to treat or prevent various diseases. It is a crucial area of research that has the potential to improve the quality of life for millions of people worldwide. The process of drug discovery typically involves several stages, including target identification, lead generation, lead optimization, preclinical testing and testing clinical trials, Drug discovery requires a multidisciplinary approach that draws an expertise from various fields, including chemistry, biology, pharmacology, and medicine. It is an ongoing process with researchers continuously seeking new and innovative ways to identify and develop effective drugs that can treat or cure diseases. Recently, we all had seen the discovery of COVID vaccine. Before concluding my remarks, I would like to convey my appreciation to the speakers. Dr. Srinivasarao Karra is the Director of Chemistry, Avilar Therapeutics, Waltham, Massachusetts, United States of America. Dr. Paul Douglas Garu, Associate Professor, Department of Engineering Chemistry, Andhra University. Sri B. Satyanarayana Garu, Assistant Professor, Department of Chemistry, Government MGM PG College, Itarsi, Madhya Pradesh, for their time and support in contributing this webinar, though they are busy. Thank you very much, sir, Srinivasarao sir, and Douglas sir, and Satyanarayana sir. Thank you very much.
madam.
Webinar
Thank you,
will be
madam.
useful for the participants to get enlightened on the topic of drug discovery. I wish you all the best. I again congratulate the Department of Chemistry for organizing such an useful international webinar. Last but not least, I thank Sri Pullela Subramaniam for technical support. Thank you, one and all.
Thank you, madam. Now, I am extremely delighted to introduce my mentor, my guide, and my source of strength. And what I am today is because of him. He is Dr. Paul Douglas, Associate Professor, Department of Engineering Chemistry, AU College of Engineering, Andhra University, Visakhapatnam. And he had 30 years of teaching experience as a teaching educator for 15 years and a chemistry teacher for 15 years. Douglas Garu has been actively engaged in research programs and published about 105 papers in various national and international journals. Also prepared 6 books and 2 book chapters. His area includes organic synthesis, heterogeneous catalysis, nanocomposite, metal oxides, CNT, and graphene. He successfully completed 1 UGC MRP. Under his guidance, about 38 scholars were awarded Doctor of Philosophy. 7 members were awarded and with MPhil, and he guided 9 MPhil projects. Now I invite Dr. Paul Douglas Garu to deliver the keynote address. Welcome you, sir.
Thank you, Dr. Sunita, for the wonderful words. Am I audible? Okay. Yeah, it's my privilege to be with you all this morning. First of all, I'd like to convey my special thanks to Srimati Kandukur Rajalakshmi Government Degree College. First time I could able to see that word, Government Degree College. Such an honor that Government Degree College. It has become a government college now. I visited the college a couple of number of times, and it's always so wonderful to meet all the people there. A lot of students and my own students. Especially thank the principal madam for inviting me to be a part of this international webinar. I thank Dr. Srinivas, faculty, Dr. Sunita, and all the other faculty members. I'm proud that at least 2 of my students are there in the same college, 2 of my scholars. And another scholar, Dr. Satyanarayana, is one more resource person today. And for that also, I'm so grateful for the organizers. Indirectly and directly, we are directly involved. And we have Dr. Srinivas Karra. Sir, I heard of you, but first time I'm seeing you. I'm so glad that you have accepted to be a key resource person this morning, and we know your inputs will certainly help us, not only me, our students, and also all the participants who are participating in this webinar. I'll be taking very less amount of time as a keynote. Uh, First of all, I thank many of my scholars who are present in pharma industry. They are the people who brought a good amount of work in drug discovery. I thank my scholars from uh, Aravindo Pharma, Dr. Reddy Labs, GVK, earlier name GVK Pharma, NATCO Pharma. These are the people who really published tremendous work in both at the international level and the national level. I thank all of them for their work. So maybe when we look for our work in the department, it is comparatively very small in nature, maybe because of the facilities that we have. But these scholars, which we have from extramural, they really did a good job in taking the name of the department through the publications and their citations, and I thank for that. So this morning, I'm so thankful that SKR College has. I think somebody's having a mm, both the audios. I think they're using at the same venue. That's why it is giving echo. At least one need to be uh, muted so that I don't get my own echo there. Okay, thank you so much. Okay. Uh, my students working on in three areas. If I watch them closely, one is they are in their drug development. Another team who are there in analytical, they are doing the analysis, drug analysis. There is another team which much focuses on uh, identifying the impurities during the process and then trying to uh, re uh, uh, prepare them and characterize them and identifying the source of those impurities. So when I look to the kind of work that we are producing, one is purely on synthetic work and their characterization and its influence or its activity on one side. On the other side, during their synthesis, what are the impurities that they are identifying? That is one work. And on the other side, we have third category, those who are doing only analytical work in terms of the drug analysis, both uh, at the at very lower concentrations and also after the metabolism. Okay, that's how I have a good exposure to this work which my students are doing from the lab. And this morning I'd like to uh, just throw a couple of lights on the drug discovery and the development. We know we used to use the word medicine for a quite good amount of time, and later on now we started using drugs, and now during the last couple of 3 years we could able to see that entire world was shut down except the drug industry and the pharma industry. This is the only industry which went through all the phases, and not only we could able to see the available drugs being used during the pandemic of corona, and simultaneously people were trying to develop within that short span of time. On one side there is a panic, on the other side there is something called as emergencies there. So on the same side, they are trying to develop new drugs for this pandemic COVID, and without uh, exaggeration, I can tell that simultaneously the word vaccine has been developed, implemented, and brought into experimental or clinical trials. We could see something the whole world never expected. Within a span of few months we could able to identify the disease, identify the cause of the disease, then trying to see how best it can be mitigated, and on the other side the vaccination part, which will be trying to stop this particular disease and That's how we could able to see a new trend. Maybe when we see the present kind of life, we know the kind of words that we listen in the present day medical field. Uh, these words were not there when we are young. We now we have so many words, so many diseases, so many things are there, and we could able to see so many drugs have come into. It has become a multi-billion dollar industry throughout the world, and we could able to see the giants in the field of pharma industry today. And if you look to this area of how this is it is actually developing, it all starts with something called as a disease. So when you are looking for a disease, then immediately some kind of unrest, unease, unease in the individual, in the family, because every kind of a small issue in related to health will bring a great change both for the individual and for the family, maybe on a large scale to the society. So that's why whenever somebody is ill, somebody is not well, we immediately look for a kind of medication for them. On one side, we have the food that is one of the basic thing that helps us to really bring down any kind of infirmity in us. But on the other side, we are depending much on the medication, much on the, and we have doctors who knows in and out of the disease, but we have parallelly the pharmacists, the pharmacy, the pharma industry, and I certainly believe it is the chemist who will be behind these medication, behind these drugs, who are continuously toiling in order to develop new diseases. So whenever we talk about drugs, we always start with a disease. Then we should know what is the source of that disease, what is that it has brought into the actual part or actual component in a human being or any other living creature. Then we are looking for what is the kind of drug or an opposite component which we can use in order to uh, decrease the symptoms, decrease the activity there, so that we can bring a relief to the individual. So this has become a normal feature. We know every one of us are aware of diseases, the effects of diseases, effects of even from a small cold to the biggest one, where people are scared. The moment they hear the name of the disease, almost all they are dead. When they go through the treatment, many of them cannot bear it, and they prefer dying. So than undergoing the treatment. So we can understand the kind of things that happen inside a body when somebody is taking a drug. Uh, that's why we always say it's uh, prevention is better than cure. So we need to be careful. We need to be very, very careful about our own health, the way that you eat, the way that we live, the way that we take the issues into our life. They all are the components of the kind of things that we are doing. So that's why I always believe that this is one which we are doing. Then on the other side, when somebody identifies a drug there, we want to see that how come that particular drug is affecting on. And I know whenever we say that so and so moiety is working, we always say that we call it as a lead compound. This lead compound is the one which has some element of activity against the chosen target. Maybe for us, we know during Corona, when whomsoever suggested something on the social media, if somebody says, "Okay, drink Jeera water," people wanted tried Jeera water. If somebody said, "Okay, drink something from garlic," they took garlic. Like we tried everything. We tried every element of the suggestions given from a normal kind of our food habits. to unbelievable drugs which have been given um, from very small doses to the very high extent. Okay, that's how we were trying on one side. Everybody was trying on the other side during the pandemic for other diseases which are already existing, like heart, like cancers, all these things. People are always working in those compounds which could be the the potential leads which can affect on the chosen target. But uh, to call them as drugs, it always requires some element of uh, their activity. So we need to synthesize these drugs. We need to synthesize these analogs or of the lead. We need to go for their structure activity relationship. We call them as SARS, and we need to find out the analogs of the leads. We need to find out the actual of the best of these analogs, which one has an effect, highest efficacy or highest efficiency. That's what we are looking there. That's why whenever people are looking for these moieties, they are looking for the pharmacophore, the structural features directly responsible for the activity. Then we want to see how we can optimize the structure there, how this is actually interacting with the target, but the one which is actually causing the component. And whenever we prepare this drug, still it will have some element of toxicity, because it cannot be accepted by the human body because it may have some other toxicity or some other side effect. That's why we always study both the components of pharmacodynamics and pharmacokinetics of these drugs. That's why when we are looking for the larger picture of this entire process, maybe we are going to listen from the experts very soon. This is how we are going in such a way that both from on one side we are looking through a normal procedure. On the other side, once it is exactly identified, maybe the root cause of the disease or the structure of the disease or the structure of uh, uh, the part which is actually affected, then we have the right kind of things. As we go on now, we even uh, reached a stage where we are actually doing a computer aided drug design that is helping us to really identify the exact, maybe the perfect, the perfect kind of moiety which can affect the site, which can affect the site. Maybe these are some of the uh, finer things that we have, and once it is identified, we are trying to study it, the drug metabolism, toxicity. Then it need to be approved by some organizations like FDA in US, and then it need to be scaled up for manufacturing. It need to go for clinical trials, and this need to be marketed, and ultimately it should reach the common man. And on some cases, these drugs are not easily developed. We even have Orphan Drug Act, where if it's a very very new kind of disease, nobody will come to manufacture a drug for it. If it is from the third world, nobody will do. If it is it is something related to a poor man, we don't find a kind of drug for that kind of disease. So it's a big thing that is happening across the globe. We have a plethora of industries working round the clock. Lakhs of scientists are working in developing new drugs, and it has become a buzzword now. Pharma industry. I still remember one of my friends who is a entrepreneur. He has a hardware shop, but very rich, very rich guy. Do you know? After the end of during the middle of the corona, he said, "Can we start any pharma industry?" Do you know, the one with crores of rupees of investment and business. suddenly he got an idea that can we start any pharma industry? Maybe people with money now wanted to have a greener industry like pharma industry. Maybe today's deliberations will certainly help us to understand how the world is looking, how the chemist is looking toward this drug discovery, and how the doctors are looking to us, how the kind of medicines that they are prescribing, and the base that they take in order to, and on the other side, how the world is looking to us. Are we giving them a life-saving drug, or are we just giving them a drug so that it makes a pharma company rich? Maybe today, as we go through this, certainly these things will come to know. And I wish the organizers for giving me this opportunity just to uh, express my appreciation for the kind of topic that they have chosen, and for the subject experts who are there. And as I follow this session, certainly it will be of a great pleasure for me to be a part of this international webinar. I congratulate the organizers, and I congratulate the subject experts, especially Dr. Srinivas Karra, for accepting. I think it's a, I don't know, sir, you are here in India or you are in US?
I'm still in US. Right now it
Okay.
is 1 o'clock.
Yeah, I know. I know. Very, very late night, midnight almost all. Thank you so much for giving your precious time for us this morning. Thank you so much. Thank you, ma'am.
Thank you, sir. Thank you for your keynote address. It is quite apt to today's topic. This is high time to think of what you have spoken, and this is need of the hour. Thank you. Now it's my pleasure to invite my colleague, Dr. Srinivas Garu, to introduce today's international speaker, Dr. Srinivas Rao Karra.
Good morning, all of you. Thank you for giving me the opportunity of introducing today's conversation. I feel it is a privilege to introduce Dr. Srinivas Rao. Dr. Srinivas Rao did his graduation and post-graduation at Government Arts College, Rajamahendra, which is under Andhra University control by that time. After this post-graduation, he received CSIR GRF fellowship and joined in University of Hyderabad under the guidance of Professor Govardhan Mehta Garu. He received his Doctorate of Philosophy, and later on he shifted to University of Missouri for postdoctoral. He is now the Director of Chemistry at Lebanon Therapeutics, Rajam, Massachusetts. He has 20 years of experience as a medicinal chemist on oncology, immunology, and CNS therapeutic area. Before joining in cellular technology, he worked at Alkermas as a principal scientist for about 1 year, and before that, he is with Serono R&D Institute and PMD Serono, working on various therapeutic areas including oncology, central nervous system, and immunology. He published over 20 research papers and was the co-inventor of 27 patents related to cancer, central nervous system, immunology, and reproductive health. He is the member of American Chemical Society Medicinal Chemistry Division. Thank you. Now I invite Dr. Kara Srinivasarao to present his lecture.
Thank you, Srinivas, for your kind introduction. Can everybody hear me?
Yes, sir, you are audible.
Okay, thank you. Thank you, Srinivas, for your kind introduction. And I also would like to thank all the organizing committee for giving me this opportunity. It is a privilege to share the podium with other distinguished speakers like Paul Douglas and Satyanarayana and other speakers. I really appreciate this opportunity. So I would like to start my presentation. Let me share my screen, hopefully.
Yes, sir, it's visible now. Yeah.
Okay.
Perfect.
Let me get my pointer too. Dr. Paul Douglas, he gave a great introduction. And I was not sure whether I should use the word drugs because considering the other drugs, people think about the other drugs like cocaine, narcotics. And in the pharma industry, it is pretty common to use the word drugs as a synonym for medicines. However, I put my title as how are drug medicines discovered instead of drug discovered.
Yes, sir.
Probably I will be shifting between these two words in between.
Yes,
And
sir.
please don't be alarmed if I use a drug.
Yes,
But
sir.
no, thank you, Dr. Paul. Now you already set the stage for me
Yes.
in describing the drug discovery process. And the properly, now I'll go into the little bit of nuts and bolts of this process. I'm starting with, this is the outline of my talk. I'll start with how are diseases caused. As you know, we all suffer from a lot of different diseases. And I will briefly describe what are the categories of the diseases and how we start getting these diseases. And also I will talk about the medicines work, how they work at a fundamental level, at a molecular level, at a cellular level. And along while after describing that, I will talk about the preclinical drug discovery process. As Dr. Paul mentioned, the structural activity relationship, computer drug design, and many other aspects of the drug discovery processes is important to identify a molecule that can be tested in humans. So after that initial preclinical drug discovery, I'll talk about the clinical development and conclude with some summary. So, if you look at the our NIH or any drug sites or anything things, the drugs are categorized they can be categorized in many different ways, but you can categorize into two main ways, like communicable and non-communicable diseases. And if you see the names and automatically it's self-explanatory. And communicable diseases are like infectious diseases, right? External pathogens like bacteria, viruses, fungi, protozoans, and worms. All these, when they invade our body, you become ill. So that is pretty straightforward in terms of the definition. Now, if we go a little bit, dig deep into the different types of pathogens, bacteria are a single cell organism which attacks our body. What they do is they release toxins, and those toxins attack the cells of our body and cause whatever the illness. For example, this strep throat and urinary tract infections, and of course TB, and also a lot of time people see stomach bug they call, and diarrhea, all kinds of diseases are caused by this bacteria. And the another one is the viruses. Virus is nothing but a capsule, and a genetic material was encoded in that shell. And now everybody knows about these viruses, and ranging from the common cold. And for the past 3 years, people saw the rampage of COVID virus. So, and now apart from that, there are many debilitating diseases like AIDS. There are several other diseases can be caused by viruses. And of course, these bacteria and other fungi and protozoans, they're also harmless in some cases. For example, we have gut bacteria, which helps to digest our food. However, when they interact with the body in a specific way, that's where the damage happens and people starts getting sicker. And coming to the other pathogen, fungi, people eat mushrooms, right? And they are also fungi. And also yeast. Both belongs to this fungi category. However, they doesn't cause any harm to us. They don't cause any harm. But if it attacks the cells, then you get this athletic foot or ringworm now, based on the round-shaped rashes you get. And another one is protozoans. So these protozoans are this type of worms, and especially malaria is caused by after a mosquito bite. And another category is the tapeworms and roundworms, which cause liver blindness and many other very rare diseases. So these are all communicable diseases. Now, what are the non-communicable diseases? The non-infectious diseases such as cancer, cardiovascular disease, diabetes, and kidney diseases, and many other diseases. I did not list here autoimmune diseases. In the autoimmune diseases, our body thinks there is some organism invaded us, then it starts killing our own tissues. So now, what are the causes for these non-communicable diseases or non-infectious diseases? And sometimes it is hereditary, based on the genetics of our body, and damage to DNA that can be caused by several ways, and that can lead to cancer, and mutations in the enzymes or receptors that can lead to overexpression of these particular enzymes or receptors, which can cause cancer. And also our lifestyle choices, tobacco, alcohol, and stress. Stress is the very, very dangerous and most important factor in causing a lot of the diseases. Probably you all know about this. And environmental exposure, such as sunlight, and that causes melanoma and many other skin conditions, and water and air pollutions. If you look at the World Health Organization's site, and these kind of NCDs kill 41 million people each year. Out of that, cardiovascular disease leads the pack. So there are all these diseases, and they are trying to invade us, and how we can protect ourselves from these diseases. Before we go into understand now what are the how we can protect ourselves, we need to fundamentally understand how the diseases are caused at a molecular or cellular level. As you all know, the fundamental building block of life is human cell. And it has a nucleus, cell membrane, and cytoplasm. And this all consists of several molecules, the polymer of amino acids called proteins. And the proteins are two different types. What could be enzymes. They are present in the cell, inside the cell, in the cytoplasm, or on the surface of cells, and they perform biochemical reactions. If you look at the enzyme catalysis, it is an amazing field where these molecules can perform very efficiently with high turnover rates and several types of reactions. And if you take one of the examples, the digestive enzymes amylase, it breaks down the carbs, carbohydrates, and starches that we eat, like proteases and peptidases and pepsin. There are several enzymes in our digestive system, and many other enzymes in many parts of the, in the cells in many parts of the body. They all can cause, they can all do different types of biochemical reactions. And the second type is receptors. They're also inside the cell and on the cell surface, attached to the cell surface. And what they do is they communicate with the cell inside the cell. When a ligand binds to this receptor, then the conformation changes and it triggers an event of cascades inside the cells. And one of the examples is thermoreceptors and nociceptors, which are on the skin. Thermoreceptors sense the temperature, as the name indicates. Nociceptors, whenever you feel pain, they can recognize the pain and transmits to our central nervous system. So these are all the at a fundamental cellular level, these enzymes and protein receptors are responsible for many diseases in a different. I will explain a little bit more on that. Suppose in healthy humans, under normal biological conditions, these enzymes and receptors behave normally because we have around 32.7 trillion cells, different cell types, and this cell is it has a very complex machinery. And so all kinds of reactions and transformations, everything going on in parallel. So under normal biologic conditions, most of the times, we don't observe anything abnormal. However, as I talked before, because of some external pathogens, or maybe within some hereditary or autoimmune factors or some other factors, what they do is they deregulate these cell signaling pathways. Because of the deregulation, and these protein or receptor levels either increase, it is called overexpression, or decreasing, underexpression abnormally. So that it tips the balance of a homeostasis of these proteins that causes the diseases. And one of the examples is, everybody knows about the diabetes. So insulin is this hormone protein. It's a very small protein, around 51 amino acids. And this insulin protein, generated by pancreas. Suppose for some reason, the pancreas does not produce enough insulin. And what happens is, the role of this insulin is take the blood sugar, and so that it can help the cells to take up the sugar, and the sugar is used for to create the energy. Whatever the energy we talk, walk, and think, and whatever we are doing, we are spending so much of energy. That energy comes from this glucose, and now when the insulin does not have sufficient amounts in the bloodstream in plasma, then you increase the levels of the glucose. And because of that, and then the muscle is unable to use the glucose because the insulin is the one which is the key mediator of transferring this glucose to these cells, which does not happen. When that does not happen, then you get this debilitating disease like heart diseases, loss of vision, or kidney disease. So now, if we can dig little deeper into at a molecular level, what happens is these medicines or drugs, right? When a molecule attached in the body, all these enzymes, receptors, they have their endogenous substrates, and they call ligands. But when these ligands bind to this active site is nothing but arrangement of these amino acids, which creates a kind of topology for the enzyme or receptor, and creates a cavity that is nothing but an active site. And this active site of this enzyme, when we block with, for example, take an aspirin, right? This is the structure of the aspirin. When we use the aspirin, it blocks the enzyme COX-1, and that how it does is it blocks this active site, like this lock and key analogy people floated a while ago. It is a simplified lock and key model. That means they are so specific. This particular molecule can go into this particular site, but not any other site, or not any other enzyme also in some cases. That's where you get exquisite selectivity. So what happens is, as you know, you cannot take a car key and open your front door. Similarly, these type of molecules, when they bind to these enzymes, and then you are blocking the active site so that the natural substrate cannot bind here, and that blocks the function of this enzyme, and which can control the disease. In the case of the receptors, what happens is when this ligands binds to these receptors, and they undergo a conformational change, and what they do is then they release the different secondary messengers into the cell, and where they can trigger a very downstream of events, and these are all chemical reactions, chemistry at every level. So all this chemical reaction takes place and releases the ultimate response is a release of a protein in higher amounts than the required. And that higher amount of protein triggers some other events, and that causes the disease. So in a nutshell, when these ligands or medicines bind to receptors, as I said, they increase or decrease the observable biological response. And one of them is this GLP-1 receptor antagonist. When a ligand binds to a receptor, and suppose you increase the biological response, that is called agonism. And the agonists like Victoza, Trulicity, these are the compounds developed for type 2 diabetes. The other side is blocking, right? As in the case of enzymes, we can also block the receptors, or partially block also, not completely block. Partial agonists, like partial antagonists, all kinds of different ways you can do with receptors. For example, this opioid receptor antagonist, that means this naltrexone, naloxone, when they bind to the receptor binding site, it inhibits the function of the receptor. And these type of drugs are used for the opioid use disorder. And suppose people are using these opioids, which are narcotics and cause a lot of problems. And in US recently, it is becoming a major drug abuse. So when these compounds are used, and then probably you can make the person normal after maybe a few years or few months time based on the medication if the person takes continuously. So this is the mechanism how medicines work at a molecular level. So now I will get into a little bit more of the how actually we do in pharma. Already Dr. Paul, he clearly mentioned exactly the processes, and now my life becomes a little easier in terms of explaining these things. So in the early drug discovery process, what we do is we select a protein that is an enzyme or receptor which causes the disease. This is I'm not going to talk about the target validation, which is a very different subject. First, for example, the COX-1 is the enzyme for the aspirin, right? First, people try to figure out how the COX-1, blocking the COX-1 enzyme with aspirin is able to block the function, whether the COX-1 is the responsible for causing this headache or whatever the other diseases. Once you identify that, then we select that particular target for developing a drug for that particular disease. And the first step actually coming to the actual process is the first you prepare this enzyme or receptor using different host systems, and then you express them, you purify these enzymes and use those enzymes, and at the same time we synthesize chemical compounds. If it is a biologics, and we use proteins like antibodies. So once the enzyme is prepared, and we also use a lot of chemical compounds to screen these enzymes. And the first step is screening of a target with a number of compounds, sometimes 300,000. In my experience, we used 500,000, 500,000 compounds to screen for one target. And that is called high throughput screening. Once you do that, out of that, based on the assay protocols, you will be able to identify the initial molecule which shows some activity against that particular enzyme or receptor, which we call hit. And then we take that hit and use the structure activity relationship. That means we try to modify the parts of the molecule. Suppose if you take this aspirin and functional groups in different places and test each molecule against this enzyme activity and determine which molecules are showing the activity. Once we identify a molecule like that, we also can use computer-aided design. That means we can get a crystal structure and then understanding the binding site of the molecule, and then use the chemicals that we use, the molecules that we made, how they are binding in the binding sites using the molecular modeling techniques, and understand what part of the molecule is really interacting with the protein. We keep those molecules, we keep those functional groups, a carboxy, hydroxy, amino, or amide, or a simple aromatic ring. We keep those individual groups and try to optimize the other areas for different properties. So that is the computer-aided design. And then, as I mentioned, this is a reiterative process. It is not like a one-time business. We make molecule, we test them, and analyze the data and again make the molecule, so that until we reach our target product profile, we call it. So we measure the IC50s, inhibitory concentration 50, EC50s in enzymes and cells. That gives us confidence that, okay, the molecules are now working on the enzymes and cells. So then we test these compounds in vivo, in animals. The animals that we use, mice, rats, dogs, and monkeys, depending on what question we want to answer. We mostly use mice and rats for the pharmacokinetics, I come in a minute, as well as pharmacodynamic effects to see the effect whether the molecule is working or not. And dogs and monkeys are for the side effects, the toxicology purposes. So we measure efficacy, how effective is the molecule in blocking the enzyme, thereby showing effect on the disease, and also what kind of side effects these molecules are causing. Once we understand the molecules in vivo, then we go into the humans to determine same efficacy and toxicity. So very briefly, I will talk about this. What is a pharmacokinetics? This is what a body does to the drug. So most of the times we administer the drugs, either IV or oral or subcutaneous. There are other administration routes also. Intramuscular, that is another way of also introduced, depending on the where you want to act the drug on. So when we take the drug and the body acts upon the drug, like we take food, right? And it is digested by breaking into different parts, and then the nutrients are extracted into the bloodstream and then goes into different cells. And that's how we get our nutrition from the food we eat. So there are a lot of chemical processes and many other events occur inside our body. The same thing happens when we take a drug. So what happens is when the drug is taken, we see the absorption, distribution, metabolism, and excretion. These are we call ADME properties. These all four things can happen for a drug. And we follow the drug concentration a certain number of hours, understand the relationship with the concentration and the time. That is pharmacokinetics. Once we understand that, we need to know that does not help in any way. But no, this pharmacokinetic helps whether how frequently we need to dose a molecule. For example, aspirin, in some cases, or Tylenol we call here, a different molecule. We need to take maybe 2 tablets or 600 milligrams, okay, 4 times a day, something like that. The reason for that is the molecules, when we take orally, they are not getting into the blood circulation like IV drug. And because of that, the availability of the molecule in the systemic circulation is very less and does not help mitigating the headache or other pain, any muscle pain that you have. So that is the pharmacokinetics tells us what is the half-life of a drug, how fast it is getting out of the system. Once we understand these factors, we also try to understand what medicines are doing to our bodies. This is called pharmacodynamics. So pharmacodynamics is the body's biological response to drugs. So whether we are seeing any effect by taking a drug, whether my headache is gone, or my wound is healed, or some kind of respiratory cold is gone, or any effect we see when we take a drug, that is the pharmacodynamic. I'm trying to simplify some of the concept. There are so much to it, but I'm trying to give an overview, a glimpse of the drug discovery and how the drugs act. So this, once we understand the pharmacodynamics of a drug, then we call PK/PD relationship. So this exposure, that means the drug exposure in response to the drug exposure with time and how the body is responding to the drug. This is the PK/PD relationship that establishes the efficacy of a drug. We can compare two drugs based on these PK/PD relationships. Suppose one molecule has only 2 hours half-life and it only inhibits 80% of the target and only gives 50% of the drug effect, but another molecule do much better, then we chose that molecule for further development. So there are many properties needs to be balanced using our chemistry knowledge. And there are of interdisciplines, as Dr. Paul mentioned, a lot of disciplines comes into the play to develop a drug. Now, once a clinical candidate is selected after identifying a molecule with all the balanced properties, now the safety and efficacy profile, everything is done thoroughly and understand the molecule, then the clinical trials in humans will start. So there are different phases of these clinical trials. And the phase 1, before clinical trials, there is exploratory studies. The first studies we do is very small scale in the lab, not under good manufacturing or good laboratory practices. Just we want to know whether the drug works or not. Once we understand that, there is a period dedicated to make the drug in large quantities under very good manufacturing practices. Even washing the reactors and everything is documented very clearly. That is declared by the regulatory authorities in every country. So once the molecules is made under good laboratory and manufacturing conditions and repeated all the studies that we do in early discovery, and then the compound is ready for going into the humans. And after the initial investigational new drug application called IND, once that is filed with the regulatory agencies, they give a green signal to test the molecule in humans. And there are different phases. Phase 1, it mainly evaluates the safety in humans, whether the drug, how much drug you can give. That's why in some cases it could be 10 milligrams or 1 milligram. In cancer case, now you need to dose until to the maximum tolerated dose, because cancer is such a very aggressive disease. So if you do 1 milligram, it may not have any effect. So you need to go higher and higher. So there are some cancer like 600 milligrams per day, or maybe sometimes twice a day. That means 1.2 grams per twice a day. So first evaluate the safety, at the same time understand the pharmacokinetics, how much drug concentration at different time points. This is done in a very small group of people, depending on the indication, 25 to 30 patients for cancer. But if it is for example, a diabetes or some other, this is not that. Though they are chronic diseases, not like a life-threatening diseases, then you need to do in a very high number of patients to get a good confidence in the molecule. And phase 2, once the molecule shows with good safety, and then if we are successful, then go to the phase 2. In the phase 2, you evaluate efficacy, as I mentioned about that pharmacodynamic effect. For example, if you take about diabetes, you use the drug and check the glucose levels, which is easy, right? Drawing the blood, but it may not be easy in many cases. you need to have a biomarker and diagnostic biomarker as well as prognostic biomarker, 2 types of markers and all those things. You need to have understanding of how the drug is modulating those levels of those markers, and based on that, you can understand the efficacy part of it. And safety. Safety is common in all the phases, even after marketing. So, and you understand that in 100 to 300 volunteers, and that the drug is showing good efficacy, and then you define the dose for the next phase 3 studies. In the phase 3, now very large number of patients, and 400 to 500 cancer. If it is a rare disease, no, maybe the number of patients may not be available. So then the number may be reduced. For example, 3,000 patients, like cardiovascular or any other bone-related disorders, for example, diabetes, they need to have at least 2,000 to 3,000 patients. After the phase 3, then the companies, pharmaceutical companies, gather all the information and submit to the regulatory agencies like Food and Drug Administration here. And I think the drugs, Center for Drugs, I forgot the, just now, the Indian Drug Agency. Like that, in every country, they have the regulatory agencies. So in one country approved doesn't mean that now every country approves the drug. So each country has their own regulations. So the data, all the data are submitted to these regulatory agencies, and those regulatory agencies, they assess the risk versus benefit with the help of the experts, and then approve the drug. Once the drug is approved, then it comes to the market. Even after coming to the market, it is possible that the drugs can show side effects if you take for a prolonged period. Idiosyncratic toxicity called sometimes. That means you may not see anything during the clinical trials, but after prolonged use, and then in certain antigens can cause immunogenicity issues, and that create problems. So in those cases, and the drug can be recalled back also in some cases, or some cardiovascular effect, which you do not see in the early stages, but after prolonged use, you may see that. So after that is the phase 4, getting more and more information even after the drug is marketed. Again, this goes by case. In some cases you need it, in some cases you don't need it. So in a nutshell, and this all costs, it's a lengthy and costly process also. The numbers change a lot, and also indication wise. And the cost of a drug could be anywhere 1 million, close to 975 million to $2 billion. And that means at least 6,500 crores to a higher limit. So overall, I hope I gave you some idea, some overview about this, how these medicines work and what causes the diseases, and what are the important fundamental building block of our cell. Now, what type of cell components that are crucial for these essential functions, and how they play a critical role in disease mediation. And as I mentioned, this drug discovery is a complex and involves several years of research and development. A typical drug takes 10 to 12 years of research, and finally getting to the clinic and everything. So with that, I thank you all for your attention, and I'll be happy to take any questions you may have. Yes, please. I think someone asking a question. Yeah. I cannot hear anything. Can you hear me? Hello.
Good morning, sir. It's really a very highly informative session. So my question is, what is the half-life for the drugs that we are using in the cancer, especially in the chemotherapy?
So it's a very difficult to answer the question in the sense that, just a second, please. Each drug will have its own half-life. And in some cases, it could be days. For example, if you take an antibody therapeutics, like Herceptin, okay, EGFR inhibitor, they may have, because the antibodies are very big molecules, they can have half-life sometimes 2 to 3 weeks, or sometimes months also. But if it is a small molecule, this small molecule will be very clearly, easily metabolized by liver, and the half-life could be maybe 6 to 8 hours. In humans, maybe a day. Okay. In some case, maybe 2 days. In the chemotherapy case, people try to do continuously because the progression of the what you call the proliferation of the cells is very, very fast. So you don't want to take any chance of giving the cancer cell another chance. So they try to treat it very aggressively every day, and then different regimens they call. For example, in some cases, maybe they give 1 week or 2 weeks chemotherapy and give rest for some time so that the patient can recover and again give another dose. Like that, they continue. Or they do the in conjunction with the radiotherapy. I hope I gave an answer which is not a what do you call a straightforward to answer. I mean, for each molecule, it will be very different. Did I answer your question to some extent?
Yes, sir. Sir, one more question. So you have told that under disease conditions, the enzymes or else the receptors are either overexpressed or underexpressed. So by using the medicine, that is the ligand, that we are inhibiting the receptor, that means the action of the receptor if it is overexpressed. So why can't we make them to have the same normal ability by means of medicine? Why we have to inhibit its receptor?
What happens is it is like a, suppose it's not like an isolated incident. Okay, these are interdependent in a cellular mechanism. These are cellular transduction pathways, they call it. So that means it is like a chain reaction going on in our cells. It is not, okay, suppose there will be endogenous substrate which bind to the enzyme or receptor, and that triggers that in turn. For example, there is a group of enzymes called kinases. Kinases, what they do is they phosphorylate the substrates, another substrate. That, if it phosphorylates one substrate, that phosphorylates another substrate. And because of this phosphorylation, they go into the nucleus and then changes the DNA patterns and then start to overproducing that protein. Okay. So now when you block it, then this, even if it can go into the nucleus, okay, and modify the translation or transcriptional aspects, there won't be enough of the protein comes out because you are stopping the natural substrate, you are blocking the natural mechanism. Okay, that. See, in normal cells, this goes very smoothly, but in cancerous cells, what happens is a mutation, a change in one amino acid in the enzyme, or that causes this aberrant proliferation. That signals that by taking this, what happens now? The cell thinks that, oh, I need to produce more of this, because there's something is wrong. Okay, because that mutation changes the dynamics of that cellular machinery. Because of that, then it keeps on producing more and more. Then you have to control that. For that, you need to put this. And in gene therapy and some other way, advanced therapies, what they do, they try to do only once, like you mentioned, right? No, you don't have to do again and again, take the molecule again and again. Okay, but that is a patient-to-patient derived medicine, precise medicine they call. It is coming up. In some cases, it's called CAR T cells. T cells, they are trying to modify those T cells and trying to, you need to take out the blood, infuse, and all those things. It is a lengthy and complex process, but there are drugs out there to treat this kind of proliferation aspects.
The last one. Why the efficacy of drug is different in different persons? I think the DNA is only the reason.
No, the efficacy is different in different person. This is a very good question. So that happens, there are a lot of factors. The composition of that person's genetic material may not be exactly suitable for this drug. Okay. And for example, if you take insulin, type 1, type 2 diabetes, right? In some cases, people get insulin resistance after some time. In some cases, they don't even produce the insulin. You cannot give the same drug both, okay, to some extent. And also, the food habits, the other risk factors, okay, and also the there are CYP enzymes called. These CYP enzymes, they can differentially metabolize the drug, okay, in different people in a different way. So there are individual constitution. That's why people try to do these clinical studies in large number of patients. It is not just for one person. Suppose out of 100, 2 people it works. That means it is not working in general. And they look at in one case, the food habits, because of the food habits, their CYP enzymes may be different levels. They are producing in different levels. Then the drug is metabolized more quickly in that person compared to the other person. Or some other genetic factor is blocking that. So the efficacy may not be the same. That's why they try to get as many people as possible, and then statistical significance. And statistically, you need to have a p-value, they call it. I do not know a lot of details, but they have a p-value less than 0.0001. That means the drug is effective in most of the population, not a few people.
Thank you, sir. Thank you so much.
Thank you, sir. You can anyone to ask questions? Audience, it is unmuted. Sir, good morning. I am Dr. Anuradha from English department. I am very much interested in chemistry, and I am very fortunate to hear you, sir. I heard often this word MSSA. What does it mean? You have also used the word essay.
Assay, okay. So now assay is a method to understand the molecule is working or not. Okay. I just told you that the aspirin, the enzyme COX-1, right? So what in the lab, what they do in the test tubes, they produce that enzyme, and they know that the COX-2, what it can do. So they use all the reagents. They have a control and use that control. See, it is normally working first. Okay, it is producing the what it is supposed to produce, maybe a prostaglandin. Okay, and once they establish that, then take this molecule, whatever chemists or make or biologists make the protein, then they simple test tube, not exactly test tube, but it's called wells, very small plates. In that, no, they add all these reagents and see the readout. Their readout is could be a fluorescence, I mean, some kind of light emission. And they check with the molecule, with the drug, without the drug. And they know the difference. The drug is, suppose if it stops the light emission, then they know that. In the control, the light is full. In the other one, no, the light emission is blocked 50%. Then look at the concentration. 50% of the drug to inhibit, the target to inhibit, how much concentration you use. And this is called inhibitory concentration. So this is called assay. That means to measure the function of an enzyme or a receptor in a cell, in a test tube. That is called assay.
Is it a
Did
new
I-
word, sir? Recently
Sorry?
coined? Is it
Sorry?
a new word?
No,
After
it is
pandemic
a,
or-
yeah, it is a, from the beginning everywhere, any drug discovery starts with that. For example, if you give drug to in the clinical test, right? That's what they do. These are all the COVID test, also one kind of test. Okay. And they take the blood and then add all the reagents and see, okay, what is there, what is not there, and based on instruments, they can figure it out. That is the assay.
Thank you, sir. You are very patient enough to answer us. Thank you so much. Please unmute it. Can anyone ask questions?
Hello. Can you hear my voice?
Yeah, I can hear.
Actually, in case of COVID, Covaxin and Covishield are applied in India. Which
Yeah.
one is suitable? And some person told Covaxin is better than Covishield. Some people told if you apply the first Covaxin, then don't apply the second case in Covishield. Is it correct or wrong?
So, apologies, sir. I'm not a real doctor. I only have a PhD doctorate, and I'm not the right person to really give you that answer. And people have to look at the efficacies, and a lot of factors needs to. I think the best thing is to consult a doctor, real doctor.
Okay, thank you. I am also interested to the postdoctorate. Can I give your mail ID and contact number?
Yeah, please talk to Srinivas now, and
Right.
yeah, he can help. I mean, he can give all the information. Yeah.
Okay, thanks a lot.
Thank you, sir. Thank you, Dr. Srinivas Garu, for your valuable presentation on the discovery of medicine. You have shared wonderful information. You have mesmerized us with your enlightened talk. Thank you, sir.
Thank you
Now
for the
I-
opportunity, and thank you all.
Thank you. Welcome. Now I invite my colleague, Srimati VBT Sundari Garu, to introduce Dr. Bassa Satyanarayana Garu.
Good morning, everyone. Thank you, madam. It's my privilege to introduce Dr. Bassa Satyanarayana, Assistant Professor, Department of Chemistry, Government MGM PG College, Itarsi, Madhya Pradesh. He is the HOD of the Department of Chemistry, who received the Best Academician Award in 2020 by Elsewhere. He completed his PhD in chemistry in 2017 under the guidance of Dr. S. Paul Douglas, Associate Professor, Department of Engineering Chemistry, Andhra Pradesh. Dr. Basta is a great academician and researcher. He published 13 research papers in reputed journals, 11 books and 2 book chapters with international publishers, and he published 7 books as an editor. He has 2 Australian patents and 2 Indian patents. He was qualified in APSET, CSIR NET, JRF for 2 times, GATE 2014 to 2018 for 5 times with All India 163 rank. He was qualified in many public service commission examinations like MPPSC, HPSC, PPSC, UKPSC, UPSC, IOCL, BPCL, NFL, ONGC, and BARC, etc. All these are the proofs of his success. Now I invite Dr. Bassa to present his lecture. Welcome, sir.
Thank you, madam. Am I audible, madam?
Yes, ma'am. Yes, sir.
Okay. Thank you, madam Srimati VBT Sundari, madam, for your nice introduction in detail. Good morning, everyone, and welcome you all. The chief patron of the today's webinar is Dr. Bhola Bhaskar sir, Commissioner Collegiate Education, Government of Andhra Pradesh. And the keynote speaker of the today's international webinar is Dr. S. Paul Douglas sir, Associate Professor, Department of Engineering Chemistry, Andhra University, Visakhapatnam, Andhra Pradesh. I am a student of Dr. S. Paul Douglas sir. What I am today, that is only because of Paul Douglas sir. After a long time, we met in same platform here. That is very fortunate, and it is a memorable moment to me and to Paul Douglas sir. And thanks to all organizers for inviting me to deliver lecture in this international webinar. And the eminent speaker, Dr. Srinivasa Rao sir, Director of Chemistry.
Your
Madam.
video is invisible.
Hello. Hello.
You are not visible, sir.
Is it visible, madam?
Yes, sir. Thank you.
Yes. Okay, madam. The eminent speaker, Dr. Srinivasa Rao Karra sir, Director of Chemistry and Avelia, United States of America. And the patron of the today's webinar is Dr. Vemuri Raghaver Kumari, madam, Principal of SKR Government College, RAJAMAHENDRAVARAM, and the organizing secretary, Dr. M. Sunita, madam, and Dr. CHP Srinivasa Rao sir, and all organizing team members of that college, and all research scholars and participants from various colleges and various institutions from all over India. A very good morning, one and all. In this short of greetings, I will start with my topic. Before start the topic, I will share my presentation. If it visible, then let me know, madam. Is it visible, madam?
Okay, sir.
Okay, thank you, ma'am. Before start my session, I will put the small quote. Start with the small quote that is every person should have a goal, should set a goal. While setting the goal, please make a proper plan. After make a plan and after set a goal, you have to continuously working hard and work smartly and thinking smart, then definitely achieve the target. That is, I want to share that to start with my session. Uh, that is my today's talk is drug discovery, drug delivery systems and their applications. Actually, the topic of today's webinar is discoveries in medicine. That's why I introduced the drug discovery. Actually, the main topic of my talk is drug delivery systems and their applications. The outlines of the today's topic is history of drug discovery. The basic, the history, what is the history behind the drug discovery? It means from the ancient times to nowadays, what the changes occurs in the field of medicine, field of drug. That's what I want to say in that point. And another one is the what is the drug delivery systems. The drug delivery system means it is deliver the drug into your body where the disease occurs. That is, I want to share. I want to cover in this topic. Another one is type of drug delivery. So, what type of drug delivery is there, and how many ways to deliver the drug into your body? That all those are covered in this point. And another one is routes of delivery. What is the routes? It means the many types of ways to deliver the drug into your body. How many types of ways is there? These all those are covered in this point. And another one is factors influencing selection of delivery. What are the factors that influence the select the drug delivery systems? These are what are the factors importance in that I will cover in this point. Polymer's role. Actually, mainly the drug delivery systems are made up of with the polymers. Mostly the biodegradable polymers is using to prepare the drug delivery systems. That drug delivery systems is used to deliver the drug into your body. That all those are covered in this biodegradable polymers. And another one is the conclusion. These all are the outlines of the my today's talk. The history of drug discovery. Actually, all of you know about from the ancient times to nowadays, what is the changes in occur in the drug discovery and medicinal field. Yeah, medications. Different types of changes are occurred. But in that, in the ancient times, the peoples are not aware about the medicines and not aware about the different illnesses and diseases. They don't know. And disease, how the disease will occur, and what is the type of disease, and how what are the medicines we will use to cure the diseases. They don't know. They don't even knowledge of about the medicines in the ancient times. But in that some ancient medicines in that time also in the ancient era. There is some medicines from Babylonian, China, Egypt, and India. India also a great play key role in the ancient time also about the medicine. And when that time the mainly focus on medicines occur from the medicinal plants. They are isolate the medicines. Medicinal plants are used for the diseases and what type of diseases occurs. They use the different types of medicinal plants and herbs to cure the diseases. In the trial and manner, they are used to cure the diseases because they don't idea about the different manner they are using. And mostly in some diseases like that is not curable diseases, they don't know. And they strongly believe that in magicians like magical people, so you go for there. Then what are the those talent that strongly believe and strongly believe some type of prayers in that times cure the diseases in the ancient times. But in nowadays, practical changes will occur, and in the pharmaceutical industry, many drugs are prepared, and or diseases. What are the type of diseases? Everything will know. But in the medicines, there is a lot of side effects will be occur in that drugs also. But in the ancient time, what are they are using in medicinal plants and herbs? That is actually that is there is no side effect because that medicine is adapt to your body already. The plants is already adapt to your body. That's why they didn't get any side effect. But in medicine, what happens is if you taken the medicine in your body, that is before it is not adapt in your body. That's why it creates some side effects. That's why that is the challenging situation. How to cure the disease without side effect, that is the most challenging one nowadays. Traditional medicine in India, actually four types of Vedas is there. In that the other one Veda, which belongs to the earlier iron age of true lights in the fact that early Indian used medications alignment from herbs and other medicinal plants. In the India, also traditional medicine also plays a very crucial role in the time from the 800 BC till 1000 BC. It is considered as a golden age of medicine in India with the introduction of medical treatise by Chakra Samhita, a physician. In that Chakra Samhita, that is the create the positiveness to patients, create positiveness and positive, generate the positive energy into the patients to cure the disease in the Chakra Samhita. And the Susruta is the surgeon of ancient India. That is the surgeon where in that ancient time, in the part of Susruta, different types of surgical instruments are there, used like metal instruments like copper, etc. In that there is the copper and extra instruments will have antimicrobial activities, anti-cancer activities. That is very useful in the time to cure the disease while by surgical instrumentation. And the Indians were known to have identified around 700 to 800 medicinal plants in ancient time also. Different plants are also in the ancient times, medicinal plants, they cure the disease by using medicinal plants. But what is the drawback? What is the drawback in that is there is no record in the data, and there is no different types of plants, what are they, and how it is useful in the correct data is not available in the present situation. That is the most drawback in the traditional medicine. If it is in the correct way, to many people in the many people, they don't know how the plants are used in the as a medicine. They don't know, they don't have a knowledge in the about traditional medicine. Actually, I hope all the persons should know about the plants and different various food items also used to cure the disease. That is the most thing to cure the diseases, I think. Actually, the main topic of my talk is drug delivery systems. Drug delivery systems means it is the deliver the drug into your body to cure the disease. Where the disease occurs, that delivery system is taken into the drug. It is also as a carrier, like a carriers to carry the drug in your body. This is the simple way to carry the drug. Different types of drug delivery systems is there, like capsules. It is a one type of drug delivery systems. It delivers the drug to our body, cure the diseases. This is one type of drug delivery. And another type is this is the syrup, like it is stored in the bottles also. That is one type of drug delivery. And capsules like tablets and syringe injections. Different types of injections also is there, intravenous injections and molecular injections. Different types of injections also is there. These all are the different types of drug delivery systems. While deliver the drug into your body, these all are the very useful for the delivery system. And another one is the routes of delivery. What is the routes to deliver the drug into your body? Different types of routes is that, like the mouth, by taken drug by mouth to cure the disease, and by inhalation, inhale the drug into your body, by absorption through the skin. It is also one type of route of delivery, and intervention through injection. This is also one type of route to deliver the drug. These are all the routes of delivery, but in this, these all are advantage and also disadvantages. But challenging is the disadvantage, minimize the disadvantage, and the drug while preparing the drug delivery systems, we have to focus on the not degrade the drug by using drug delivery system. That is also one of the main factor. And another one is the drug delivery system will not create any side effect in your body. These are two are more important factors while preparing drug delivery systems. It is challenging one, and nowadays new techniques also coming to the medications while preparing drug delivery systems. That is the most important, and that is factors influencing the selection of the delivery route. These are all the factors that influence the drug delivery. That is, the first one is the drug physicochemical properties. This is the most important factor while preparing the delivery systems. We have to focus on the physicochemical properties of the drug. And another one is what is the molecular size of the drug. That is also most important factor. We have to consider this. And third one is half-life. Already Dr. Srinivasan sir told that about half-life of the drug. Every drug will have a different types of half-life. He already said in the previous lecture. And next factor is the chemical stability of the drug. That is also most important factor while preparing delivery systems. We have to focus on this. Keep in mind the chemical stability also. And the fourth factor is the loss of biological activity in aqueous solution and protein denaturation and degradation. These are the most important factors. Keep in mind, and in pharmaceutical industry where the drug delivery system is preparing, they are all focus on keep in mind, and these follow these factors to while prepare the drug delivery system. Role of polymers in drug delivery systems. Actually, polymers play a key role in making a drug delivery system, especially biodegradable polymer. Actually, polymers are different types of polymers is there, but biodegradable polymer is widely used in the prepare the delivery systems because biodegradable polymer is like a proteins and peptides. In that, these are already every person needs peptides and polymer proteins. It is easily degradable, biodegradable in your body. That is the most important thing. If it is biodegradable, that means there is no side effects will occur after the giving of the disease. That's why biodegradable polymer plays a very key role in drug delivery systems. In the pharmaceutical applications, actually in the preparation of the tablets, there is a biodegradable polymers which are the binders in the tablets and controlling agents in the liquids and emulsion suspensions. Polymers can use as a film coating in distinguish mass unpleasant task to drug, enhance the drug stability to the modify the drug release characteristics. While in the pharmaceutical industries, polymers, what the polymers plays a key roles. These are that's why these polymers have a very vast applications in the pharmaceutical industry to prepare the drug and even drug delivery system also. The biodegradable polymers, already told about biodegradable polymers. The biodegradable polymers have a wide applications in the biomedical applications because the biodegradable polymers have a bioavailability and biodegradability. Of this biocompatibility and biodegradability, it have a vast applications in the biomedical applications. And another one is these biodegradable polymers is the more efficient and minimize side effects to other types of inconvenience for patients. That's why these biodegradable polymers have a wonderful scope in the pharmaceutical industry and the medicinal industry and the drug industry. Coming to the next slide, that is different types of biodegradable polymers and their applications. Many types of biodegradable polymers are available, and in that the one biodegradable polymer is cytosine. This is biodegradable polymer. In this cytosine, the monomer unit of cytosine is glucosamine and N-acetyl glucosamine units. These are the monomer units of the cytosine. It is obtained from the deacetylation of cytine, derived from the exoskeleton of the purustine. And the cytosine is valuable properties as a biomaterial because it is constituted biocompatible, biodegradable, or non-toxic. Because of these factors, that is biocompatibility and biodegradability, that's why the cytosine is widely used in the biomedical applications as a biodegradable polymer. This also has a cationic character and functional potential functional group. That's why this is have a vast application in the biomedical and pharmaceutical applications also. And cytosine also has been used in many formulations. Like powders, tablets, emulsions, and gels also because of these character like cation character, etc. The structure of cytosine is this is already know about the structure of cytosine. This is like one type of polymer of the carbohydrate. Everybody knows about carbohydrate. Everybody needs carbohydrate also. Everybody taken daily carbohydrates in the different from the different food items. This is needs your body also support. If it is taken this cytosine as a drug delivery systems, it easily taken the drug into your body, and after curing the disease, it easily degrade automatically. The carbohydrate system is already adopt your body. That's why there is no side effect is getting by using this and. Sorry for the interrupt you. The power is gone. And another thing is this is after this is the biodegradable polymer. It is mostly different types of polymers is also is there. That is starch, gelatin, gene, and different types of biodegradable polymers are also available. Once again, I will share my screen in this mobile also. Am I audible?
Okay, sir.
Yes, madam. Light was gone. That's why. My screen is visible or not?
Not visible, sir.
Is it visible, madam?
Your screen is not visible, sir.
Now?
Okay. Okay.
Yes. Okay. The another biodegradable polymer is cellulose. It is a well-known biodegradable polymer. Everybody know about cellulose. The monomer unit of cellulose is glucose. It is also a one type of biodegradable polymer used as a drug delivery systems. In this, it is hydroxyethyl and hydroxypropyl cellulose are mostly used as a biodegradable polymer. These hydroxyethyl and hydroxypropyl cellulose are soluble in water and alcohol also. For it is mostly used for the tablet coating and tablet matrix and gelatin and capsule materials, etc. The structure of the cellulose is here mentioned in this slide. It is also the polymer of the carbohydrate. It is easily biodegradable, and there is getting the no side effects by using these drug delivery systems. And the another one is rosin. It is also a one type of biodegradable polymer. Rosin is a film forming biopolymer. It is mostly used in the cosmetics, chewing gums, and dental varnishes, and prepared spherical microcapsules, etc. And another one is the gene. It is also a one type of biodegradable polymer and alcohol soluble proteins as a byproduct. And it is an inexpensive and most effective substitutes for the fast disintegrating synthetic and semi-synthetic film coatings. This is also used for the edible coating for foods and pharmaceuticals also. And another one is the hydrogels. Hydrogels, it is means it is hydrogels as the cross-linked hydrophilic polymer. Actually this is imbibing the large volume of water but insoluble in water, because it is the very complex structure, very network structure. That's why hydrogels are not soluble in the water. It is also used as a biodegradable polymer for the saccharides, and useful for the markers to monitor several physiological changes as well. And the another one is liposomes is the liposomes is the most important and most useful biodegradable polymer in the drug delivery systems. It is the most important one. In this, liposomes are created from the cholesterol and natural non-toxic phospholipids. These are the showing particular promise as a intracellular delivery systems for antisense molecules, ribosomes, proteins and peptides, and DNA. Liposomes with enhanced drug delivery to disease locations by ability of long circulation residence times are now achieving the clinical acceptance also. And another biodegradable polymer is the nanogels. Nanogels means it is the material, that is the particle size is in the nano, that is 0 to 100 nanometers. Nano materials is also used as the drug delivery systems in present technology. It is most important drug delivery systems. But in the field of in the some drug disease like in the brain area, but brain is very sensitive area where the nanoparticles as a drug delivery system is created some problems. But in the future challenging scope of drug delivery systems in the brain is also future challenging task area to prepare the most important drug delivery systems while disease during the disease in the brain area. That is the most important and challenging task also. In the and the some others are starches. It is deliver the proteins or peptidic drugs orally, microcapsules containing a protein. Pectin is also here. It is possess the several requisite characteristics to be used as a polymer in drug development and release kinetics. Gelatin has been used almost exclusively as a cell material for hard 2-piece and soft 1-piece capsules also. The conclusion of the talk is various polymers are used in biological and biomedical applications exclusively in the significant drug delivery systems evolution. The main goal for developing such delivery systems is to minimize the drug degradation and loss, to prevent the harmful side effects, and to increase bioavailability. Thank you so much for giving this opportunity to deliver the lecture. Thanks to all organizers and organizers also. Thank you. If anybody asks the questions, you can ask.
Any questions from audience side? Okay, thank you, Dr. Basta, for your valuable presentation on drug delivery systems and its applications. Your lecture is very useful and informative to the upcoming generations. Thank you.
Thank you, madam.
Now I invite my colleague Srimati N. Swati Garu to introduce to present the vote of thanks. Good afternoon, everyone. On behalf of Department of Chemistry, SKR Government Degree College for Women, I am glad to propose vote of thanks. I thank our Honorable Commissioner of Collegiate Education, Dr. Paula Bhaskar Garu, for his encouragement and support in organizing this webinar. A special thanks to our chief guest for accepting our invitation and being a part of today's webinar. Thank you, Dr. Paul Douglas Garu, for sharing us some insightful knowledge on the difficulties in drug discovery and how to overcome them. Thank you, Dr. Srinivas Rao Garu, for your valuable presentation on the discovery of medicines. The discovery of medicine is a critical step in the development of new treatment for diseases and can have a significant impact on patients' health and quality of life. You have shared wonderful information. You have mesmerized us with your enlightened talk. Thank you, Dr. Basa Satyanarayan Garu, for your presentation on drug delivery system and its applications. Drug delivery system has a potential to improve drug efficacy, reduce side effects, and enable targeted delivery of drug to specific tissues or cells, leading to better treatment outcomes and improve the patient quality of life. Your lecture is very useful and informative to the upcoming generations. I thank Dr. Raghu Kumari Madam, our principal, for her cooperation in organizing international webinar. It is always a privilege to work under her leadership. I hope to feel her proud to our future endeavors as well. I also extended thanks to all staff members for their enormous cooperation in the organization of this international webinar. Thank you. Madam, video off this.
Okay, bye everyone.
Thank you, sir. Thank
Thank
you.
you, madam. Thank you all.
Thank you all. Bye bye.